Hello, and welcome everybody back to TOXstream, the live streaming show of AlterTox Academy, which brings you the voices of experts on toxicology and alternative to animal testing. My name is Matteo, and I'm the host of the show. And I'm very happy to be here, and I thank you everybody for joining and tuning with us tonight. I hope you're going to have a good time with us. So for the people that don't know us that well, here at AlterTox, at TOXstream in general, what we do is promoting and disseminating alternative method to animal testing as best as we can. We have a wide range of activities that we do. We are very active on social media. We produce podcasts. We have this live streaming show. We even have our own board card game to explain to people how to create new approach methodologies and new alternative method to animal testing in toxicology. And we also collaborate with different EU projects that are somehow related to toxicology and NAMs. If you are interested in learning a little bit more of what we do, you can check everything on our website, altertox.academy.be. There you can find all the different things that we do, all the different activities, the links, everything else, and also our contacts in case you want to reach us and contact us. While TOXstream, this show, well, as I said, this is a place where we pick a topic always, rather either on toxicology or on alternative to animal testing, or science, a little bit broader sometimes. And then we try to find the best speakers, the best guests as possible that can help us understand a little bit better this very complicated world. And this is what we're going to do tonight, as we did already a few times, because this is indeed our 12th episode. So we already had a few. We started back in February, if I'm not mistaken, or maybe March, I don't know. But you can find back all our past episodes on our YouTube channel. Probably that's the easiest way to do it. You can either go on YouTube and type AlterTox, or you can also check again on our website. Everything is there. And right now we are live on YouTube, LinkedIn, and Twitch. That's how we usually do it. And the fact that we are live, it means that we are also giving you the chance to interact with the guest and the expert that we bring online. Because indeed, you can use the chat. No, doesn't matter which software you're using to follow us. You can use the chat. You can just type and write your comments, your question, your suggestion, whatever you have in mind. And I will do my best to bring it to attention that of our expert when possible. So with that out of the way, I think we can dive in the topic that we have prepared for you tonight, which is kind of a continuation of what we did last week, a couple of weeks ago, which was the first episode on EU project. A couple of weeks ago, we started talking of OnTox project, which is a project where they are trying to create new approach methodologies, new NAMs to replace the use of animals in toxicology. And the project we talk about tonight is PrecisionTox, which is related to OnTox mostly because they are both part of the same cluster. They are kind of merged together in this super project together with another project called RiskHunter, so PrecisionTox, RiskHunter, and OnTox in this ASPIS cluster. And PrecisionTox is indeed the second project in this list. And similarly to OnTox, also PrecisionTox have been founded by the Horizon 2020 Research and Innovation Program, meaning that it got funding from the European Commission, from Europe, to try to achieve this very, very ambitious goal of finding new ways to protect us from toxic chemicals. And again, similar to OnTox, also PrecisionTox is kind of a huge project. It's pretty big. You have 15 different partners, 15 different institutions, public and private, merged together, mostly from Europe, a few also from the States, similar to OnTox again. And the budget that they are trying to use, they manage all together, is of around 20 million euros that stretched in 4 or 5 years. I don't remember right now, but it's pretty impressive. And I'm always amazed when I see all these projects, these huge projects that are trying to put together so many different expertise, so many different institutions together, because it must be a nightmare to coordinate everything. I mean, this is basically what happened the last time that I had a tenant meeting in my building, and the only thing we had to do there was just to decide which sort of lights to put in the garage because we're missing one. And after 2 hours of discussion, we didn't even manage to decide that, even though a light is just, I don't know, 5 euros at Brico. And here we have a project where they are managing 20 million euros, which is, I cannot even fathom how complicated this might be. But PrecisionTox made it work, and we're going to talk about that a little bit tonight. Scientific-wise, what PrecisionTox is trying to do, it's also a very interesting approach, where OnTox was trying to go mostly for in vitro and in silico techniques to find new methods to avoid the use of animals in toxicology. PrecisionTox is going to use these very particular and weird animals that are now on screen, which are considered different sort of animals than the one that we use usually in toxicology. And they are very interesting for a couple of reasons. The first one is that they all look like some sort of Lovecraftian freakish horror thingy that are gonna give you nightmare if you stare at them too long tonight. And the second one is that, as I said, they are called non-traditional species or non-sentient species as well. And we will try to understand a little bit better tonight what these mean as well and why they are useful. And what PrecisionTox is trying to do is to link them in an evolutionary point of view and trying to see the way in which these animals react to toxic chemicals and link these back to human and the environment from an evolutionary and genetic point of view, which sounds extremely complicated, and that's why tonight we're going to try to get a little bit of a clearer picture of this. So for all these reasons, tonight we're going to try to give a closer look to PrecisionTox, how this project is working, what it's trying to achieve, and why it is important for all of us. And as usual, I'm not going to be the one that does most of the talk because I know absolutely nothing about it, but we have 3 wonderful guests and 3 wonderful experts that they will give us their ins and outs of the project. So I think for you, it's time to meet the expert. We have some issue with the jingle, but doesn't matter. Hello everyone.
Hello, Matteo.
Hi. I'll introduce you one by one the way in which I see you in my screen. Starting from John, the first one that I see, Professor John Colbourne. So he's the professor and chair of environmental genomics at the University of Birmingham and the coordinator of the PrecisionTox project. With too many other titles that I'm not going to put them here, otherwise we're going to spend half an hour just like that. But thank you very much for being here.
My pleasure.
The second one is Dr. Gaëlle Hayot. I don't know if I am supposed to leave the T away or not.
Excellent.
Sorry about that. Postdoc at the Institute of Technology of Karlsruhe, something like that, or something that is supposed to sound like that. And also part of PrecisionTox, of course. Welcome.
Thanks.
And the third one, boy, that's a long name. Christina Cramer von Clausbruch. Maybe.
Well done.
Okay, thank you very much. Same institute as Gaelle. I'm not going to tell it again because I already butchered it the first time. PhD student there and also part of PrecisionTox project. So welcome as well.
Thank you.
Okay, so thank you very much all for giving me one of your hours in the evening. I know it's not the best timing for everyone, but we're gonna try in this hour to understand a little bit better what you're trying to do with PrecisionTox. But before jumping in the project, I always like to ask my guests rather how they ended up doing what they are doing right now. So which is the path that they took in their life that send them where they are right now. So I think I'm gonna do the other way around this time. So I'm gonna start with Christina, and then Gaelle, and then from John. So Christina, how do you end up being a PhD student working in PrecisionTox?
Very shortly, coincidence and then interest. Because coincidence that it went to biology and not medicine, because the acceptance for medicine came later and I already was in a practical course and biology got its hooks into me and I decided to stay with biology. And then I worked with fungal toxins and switched to cytotoxicity out of interest, which landed me in Karsten Weiss's lab. And I did my master thesis with him. And now I do my PhD with him and produce PrecisionTox. So coincidence and then just interest for the topic.
Well, you managed to land in a very interesting project. So
definitely
you had luck. Okay, so Gaelle?
So for me, it was also kind of lucky, I guess. So I initially trained in genetics. Then I did the PhD on zebrafish already, studying the genetics of autism spectrum disorders. And just when I was looking for a new opportunity, I saw the advertisement for PrecisionTox, and I really liked the project as it was shown. So I really wanted to be part of it. And here I am.
Cool. So you really went for PrecisionTox. Okay, that's interesting. And John, how you ended up in here?
Well, I ended up doing toxicology through a different route than most people. I came about toxicology because of my appreciation for the role that a model species called Daphnia plays in ecotoxicology. So one of your crazy critters that you were showing earlier on is the water flea Daphnia. It is a crustacean. It's found in all the freshwaters around the world. And as a youngster, I would canoe on these waters in Northern Ontario and capture these critters and ask, what got you here? Why is it that you're able to actually survive in this environment? Sometimes the environment is very nice and you can understand why. Other times the environment is totally polluted and you still see them in those environments. So I was very curious as to find out how is it that an organism or a population can survive very toxic environments, which led to understanding a little bit more about biology from the studies of these model species that can tell us something about biology in general, including evolutionary biology, but in this case, also being very applicable in toxicology because it is the model species for setting regulatory limits on what compounds are allowable in the environment based on studies using Daphnia.
Okay, and I know that this is indeed because you are mixing not just biology, but you also have toxicology, biology, and environmental toxicology at the same time, which is a very interesting take. But I see, I can picture you at least canoeing somewhere and having this discussion with yourself and seeing how you ended up in here. Okay, that's a super interesting story. And related to that, so PrecisionTox, as I said, like OnTox and most of the toxicology project that we have right now, are very focused on these, what we call NAMs, methodologies, alternative to animal testing, whatever you want to call them. And is this something that is also quite important in PrecisionTox in a way and in toxicology? So I'd like to ask you also if the fact that now a lot of work in toxicology is focusing on NAMs had an impact in what you are doing, or you wanted to end up in these, or you ended up working with NAMs kind of by chance. And this time, maybe we go the other way around. So I start with John, then we do Gaelle, and then we do Christine.
Oh, interesting question. A lot of people ask me whether I believe that NAMs are truly new in terms of approaches, methodologies, because in my whole scientific career, I've been doing NAMs from the very beginning. NAMs is really a technological advancement. It's using different types of data to tell us something fundamental about the questions that we're asking. Since 2007 or something like this, when we first started sequencing the Daphnia genome, at that time it was new. We were actually seeing how the application of genomics or metabolomics can reveal something very, very new about biology. And it's been transformative. It's been revolutionary in so many different fields. And so the technology is quite mature. And if you ask the young career investigators whether this is new, they would say, heck no. I know nothing but this kind of technology, like Christina and Gaelle here. So why are we calling them new approach methodologies? I think that it's simply because despite the evolution and revolution that it's actually occurred in medicine and other fields, it has not yet penetrated into regulatory toxicology for good reason. Regulatory toxicology is very much needing to make decisions based on facts and observations. And for many years, of course, those observations were very much based on seeing adversity firsthand, to actually see an animal suffer because of exposure to chemicals. But just like in medicine, where no longer do we need to describe diseases based on seeing a tumor, we can actually define diseases based on knowing something about the molecular processes that have led to that tumor. Mm-hmm. And so it's new in the sense that toxicology, ever since 2000 or something like this, has begun to actually appreciate the fact that it could be understood based on mechanisms and understanding the chemical modes of action and making decisions around that kind of data as opposed to data of adversity itself. So I think that going forward, I see that being a toxicologist today, especially if you're entering into science as a young investigator, is very exciting because we expect to see the same kind of revolutions that we've seen in so many other disciplines where 20 years ago, 25 years ago, it was indeed new. But here what we're trying to do is that we're trying to apply now some rather creative ways of applying these technologies in a brand new regimented discipline that we call toxicology.
Yeah, no, it makes sense. I mean, I really love that every time that I ask this question to people, they always give me different answer, because indeed it's such a big and large topic that is not so easy to frame it. But indeed, it is a revolution, and it is nice to ask to Gaelle and Christina if your experience was more like, yeah, it's not new. That's just how we do things today, or if there was also a little bit of changing. Maybe Gaelle, you had a little bit more experience. Maybe you did see something, or how it was.
So actually, no, when I started to train, for at least for the zebrafishes embryo, it was already very well implemented, and I would say already quite old to work on the zebrafishes embryos. So yeah, to me, that's not really new approach methodology, but more, as John said, it's more that it's now beginning to make a break into regulation. But at least in research, fundamental research, they have been used for a very long time, and they are very instrumental in finding new ways of curing people, assessing toxicology, and so on.
Yeah, no, I agree. I understand. And for you, Christina?
Yeah, I'd agree as well. They're not new to us, since that's basically what we've been trained on and what we've been doing basically since I started my bachelor. So
Okay.
nouns are more used to me than if you now ask me to go into animal testing. I would know nothing of how to do that. So we learn those as the toolbox with which we enter into PhDs and further research work. And I think it's just about finding new ways of using them to, outside of research, use them for like the 3 R's: to reduce, to replace, to refine where we can't yet use them, and to get them more out there, because it's nice that we all learn them at university and in research, but they're not yet out there in legislation. So that's where they're going as something new.
Yeah, no, I agree. I mean, maybe we should create a new term right now, and we're going to call them, I don't know, proper approach methodologies, and then we're going to call them PAM, and everybody's going to use them. Also have a bit more power than NAM, maybe. But no, it's indeed, it is cool. And it's true that maybe outside, when people hear toxicology, if you are not in the world, they really sneak probably quickly to animal testing, but this can be no further away from reality. Okay, now we can jump a little bit more into PrecisionTox work real. The question that I asked last time for OnTox, and I'm going to ask it again for PrecisionTox, is how the project came to be. And of course, this is going to be more for John. But before maybe coming to John, we can pass from Christina and Gaelle, and rather asking you how you ended up, like the first, I don't know, few weeks, few months in PrecisionTox. How long it took you to kind of understand the impact of the project, what the project was trying to do from your side, of course. We can start from Christina, Gaelle, and then we ended up with John.
Okay. So starting in the project was pretty much getting dunked into it head first, because I had, I think, 2 weeks of my PhD, and I was already ordering cells and starting the first rounds of testing. How do we need to seed them? What densities do we need? How does this work? How can we order this? How do we do this? Understanding the project, basically the first couple of meetings where we were talking about how it's going to progress and go on. I mean, I joined basically a year into the process, so there had already been a lot of discussions and decisions on how to take things and how to develop things. And then, yeah, when I came in, we basically started with the first SOPs and how do we actually now do this, and started. And then the pilot happened, and then it was like 3 months later, and I was like, okay, we're doing PrecisionTox.
Okay.
So that was my go at it.
The worst way to start a PhD, I guess.
True.
And Gaelle?
Yeah, so I actually started exactly at the same date as Christina. So for me, it was also the same. I started almost directly with the experiments, and I already had a good idea of the project because Thomas and Carsten, so Christina and my boss, explained it to me before I started. But yeah, I think it's really doing the experiment and taking part in the first meetings of the consortium that really helped me to really understand what was PrecisionTox. But it was quite quick.
Okay.
But that was very well explained also. So.
Okay, thank you for that. So John, how did you ended up with this idea? How was the process? I mean, okay, I have the starting point. I have you canoeing. Okay, how then you ended up with the proposal of PrecisionTox on your desk?
It's so wonderful to hear Christina and Gaelle speak about their entry into the project, but it does have a long history. It goes back to around 2010 or something like this, and it's born out of comparative genomics. So at that time, we began to see the genome sequences of so many different species. You would have called, well, you had earlier called them non-traditional, maybe in toxicology, but these species were very traditional in medicine, in genetics, in developmental biology. And so we're talking about having seen for the first time the genome sequences of Drosophila, melanogaster, the fruit fly, C. elegans, the nematode worm, of course, the vertebrates like mouse, humans, and Daphnia was the first crustacean genome to have its genome sequenced. And at the time, it was quite fantastic because it was a community-driven project. There were no experts in this field. So Gaelle and Christina entering into genomics at the time when I had, we were the best in the world because there nobody else had ever done it before. And so it was very exciting that way. But what we ended up doing, of course, is that through that process of learning something new about the basis of animal life, we were training ecologists on being ecological genomicists. We were training traditional fields and adding genomics to that field. And every time that we would do this, we would be creating a new field. And now we have, for instance, in ecology, all kinds of journals that make a living out of publishing genomic discoveries made in the field of ecology. So in this community-driven approach to doing science, it became quite apparent that we can learn so much more by comparing the results that we're obtaining from all these different species. And all kinds of discoveries were made there. We're not so different from one another. I mean, at the very beginning, there was always a notion that humans were special because we have so many more genes than other animals. Well, that paradigm fell apart pretty quickly. Daphnia has many more genes than human does. And so the idea of complexity in biology was no longer linked to these preconceived notions of what it means to be an animal. So as we learn more about the genetic basis of animals, the more we appreciate how common we are as opposed to how different we are. And in medicine, what that has translated to is simply the understanding that those genes and those pathways that are most tied to human health are the genes and the pathways that are most conserved across animal species. And it makes a lot of sense, Matteo, because if you're going to perturb biology with chemicals, right, and it's going to actually have an adverse outcome, it can't be genes that are responsible for the length of your arm hair, which is very much of a mammalian feature. It has to be something more fundamental to that. It has to be things like cellular biology going awry, reproductive biology going awry. As it turns out, those are the things that we as toxicologists care about. We want to protect life from those kinds of adversities. And as it turns out, the pathways that are responsible for those traits are in fact very, very ancient. So the beginning of PrecisionTox was born out of PhyloTox, to use a comparative approach in genomics now into understanding perturbation biology, to understand toxicology. And again, starting from a grassroots, we had to identify laboratories that were specialists in each of these model species. So that's how we got to know the fine folks at KIT, at UFZ, at Indiana University, and other places where we had the world experts in these model species. They can actually do experiments very, very readily and high throughput, and have access to some of the best deliverers of omics data. And by coming together in order to pursue that science, we were doing things by borrowing money from existing grants, and we were trying to kind of like crowdsource our way through this. And then voila, there happens to be a very well-constructed call for new approach methodologies. And it's not like we had assembled this team of people just to kind of like win this money. We were already doing the science, we were already doing the work, but now we had the opportunity to do it right and to do it at scale. And so I'm quite pleased and quite grateful that the European Union has decided to invest in this area of research, because it will actually lead to those fundamental discoveries that we've seen before in the new light of perturbation biology. But at the same time, people in the consortium, they tell me, John, I really have joy getting up in the morning because I know that the work that I'm doing is actually going to have significance. And just like this kind of data and this kind of approach is saving lives in medicine, we expect this to be saving lives in toxicology as well.
Yeah, no, that's a wonderful way to describe it, and I like the fact that it seems almost like a natural thing that this project came to be. It has a life on its own almost, and it's just a natural thing that it became what it became so far. And kind of related, I mean, there is this comment that I like from Martin that indeed these technologies can have the predictive technology can have really the power to help us in the 3R mission, which is, as I think Christina said before, this or Gaelle, replacement, reduction, and refinement with the idea of using less animals in toxicology. And maybe just to go a little bit deeper on that, John, so the reason to use these 5 different animals, I understand Daphnia because you've been falling in love with her in the past, but it was just a omics reason. So because we had a lot of information on the genomic level, or there was also other reason by choosing them and not something else?
It's a good question. I also appreciate Martin's comment. Martin, who gave us a comment, is very much a key player in precision toxicology as well, delivering the omics, the metabolomics data that is so critical to the project. The choice of the model species very much lies in the fact that they are models already. So everything, or most of what we know about genetics, comes from studies of fruit flies, and a lot of things that we know something about development comes from nematode worms. So they're already fundamentally good in terms of that translation between observation in the laboratory and something meaningful for animal life, including humans. But of course, they were chosen at the time because they have small genomes. And so when it required, hundreds of millions of dollars to sequence a genome, the smaller the better. And so they were organisms that were chosen because they had huge communities behind them, but they also had small genomes, and those genomes we were able to actually do something with them. But nowadays, there are some projects now, even here in the UK, where they're trying to sequence every single organism in the world. And the technology is that mature. We will one day basically have the DNA fingerprint of everything that we can see in nature. And so the designation of these as being model species is a convenience of the history of the science. But there's no reason why any one animal has value in terms of understanding that link between biology and health and exposures to chemicals. But we do have to choose things that are readily amendable in a laboratory. You can have many numbers of them growing simultaneously. They're small. You can actually manipulate hundreds of them at a time. So all of these attributes are really important, especially when you're tasked with exposing all of these models to 250 different chemicals and sampling their omics across different concentrations and different time points. I mean, really, we're talking about somewhere around 21 million, data points on the omics side. Sorry, did I say million? 21, not that many, 21,000.
Okay, yes, it's a lot, but okay.
still a lot. And so the choice of model species is critical, not just for the biology, but for the practicality of getting the work done.
Yeah, no, I understand, and it makes sense. You don't want to invent a wheel all the time. I mean, you use C. elegans from so long to do so much stuff that it makes sense to use it also in these contests. And another question still on the project itself, and you kind of said it already. The idea is, we want to study these animals because they are useful, but then of course we want to link everything else to human. We want to link everything to the environment, and we can because a lot of genes are conserved, as you said. Even though I'm wondering how easy it is, this link. Especially since, a lot of these animals, sometimes they even have too many genes. I know that a lot of them have duplicated genes at some point, so they have more copies than what we actually have. So I'm wondering how easy it is, the translation afterwards. And we start with John, and then I'm going to ask to Gaelle and Christina, because you both work with the in vitro aspect. So I will ask you also more how it is the practical aspect of working with cells and interface yourself with someone that maybe is working with Daphnia and with something that doesn't have a liver, and then you're like, yeah, but I'm using liver cells. And I'm interested to see how it is. So I'm starting with John. Just tell me how it is easy in your idea to connect this with humans and environment at the end.
I don't think it's very easy at all. Thus this opportunity to find a solution. It is a research and innovation project, and the majority of the hard work will be in terms of the interpretation of the data. Gratefully, there again, there's been some revolutions, especially in around the computational work of applying machine learning and artificial intelligence, especially those forms that are explainable. And it was simply a matter of finding the right people to try to apply these methods on this data set. So we do not know whether we will succeed. There's no guarantee here. We do know that we'll actually make some phenomenal discoveries, but whether the phenomenal discoveries will actually lend itself to being able to make an observation in a suite of model species or in cell lines and to directly make that link to adversity in humans. Gratefully, we have 4 more years to do that. So,
well, only the difficult things usually are very worth to be doing. So I think it makes sense that you're trying to approach this very complicated topic. And Gaelle and Christina, how it is from your point? Because indeed you are the in vitro part, so the human part, let's say, of the project. So I'm wondering how it is the way in which you have to interface with all the other part of animal, the people that study the C. elegans, the fruit fly, or on the other animals. Let's start with Gaelle.
Yeah, so I think that for now we are mainly comparing our results to see if we have some kind of consistency between the different species. And from what we've seen so far, it's quite good. So that's already a relief. And then also, at least for zebrafish, so we don't have always direct correspondence between human and zebrafish, for example, but the pathways are usually conserved. So even if all the different steps in the pathway are not conserved, the pathway as a whole is conserved. So I think that this is something that is possible to actually compare at the different species, although it will be probably very complicated. But I have faith in PrecisionTox. So
we all are. We all have. Christina?
Yeah, so we are currently mostly comparing. So while Gaelle is doing the zebrafish, I'm doing the cell work. And you are right, it leads to very interesting discussions about how does cell culture work, to explain someone who will do the omic samples of what is a batch when we're talking about cells compared to some species that lay eggs. And that's obviously a batch. That's not how it works with cells or with some other species. But those are interesting, and that's also just part of the fun of getting each other's language and how to even compare. And in the comparisons, I think we are doing well right now. We're seeing some things that make us hopeful. We have, like, if you're just ranking toxicity, there are similar patterns that we're very interested in and looking out for, and we'll see where that takes us. But yes, we are not guaranteed to succeed, but we're working on it.
We are very much looking forward, indeed, to that results. There is a nice comment and question from Tanya asking, what about the animals that have been genetically modified and the models that are produced with human augmentation? Will they be taken into account in study for toxicology? I guess human augmentation would be, I don't know if it's more organ on a chip or this sort of technologies, because it's something that we didn't touch upon. Indeed, a lot of these animals, you can also do a lot of modification on the genome, which helps these sort of studies. But maybe John, you can tackle this comment.
Oh, Tanya has a very good question there, and in fact, it is part of precision toxicology. Tanya, these models do indeed have a lot of reverse genetics being done on them. In Bloomington, Indiana, where they have the Bloomington Drosophila Stock Center, they have most of all the strains that have ever been used in research. Or at least that's what they're trying to collect. And so any investigator could go and order these strains and that have been modified and to test their hypotheses. And Tanya is correct there. Human augmentation is putting human genes into animal cells, for instance.
Okay.
Or even putting human genes in whole organisms, which is very possible with these models. And they are very important because for us, that's the smoking gun. In genetics, there's this need to demonstrate causality to the level where it's sufficient and it's necessary. Those are two very high standards. And in order to do that, you have to basically manipulate it to the point where you predict that if you knock out this gene, you will have that phenotype. And so in biology, that would be called a smoking gun. And so everything that precision toxicology, what we've talked about so far, is very much correlations, looking for these associations, strong statistical associations, but no smoking gun. But in work package 4, and I hate to say work packages in a public meeting like this because that's so EU talk, but there is a whole series of investigations that are doing precisely that. They're digging down into now looking at the causal links tied to variation. So there's two very interesting projects going on. One of them is that there's a genetic resource panel for Drosophila, and it's a whole population that's being exposed to these compounds and measuring sensitivity to the harmful effects of the chemicals based on genetic variation. And that variation is being checked against the same kind of panel, but human cell lines that have a lot of the genetic variation in humans, and asking the questions, are these the same genes that are responsible for whether or not you're susceptible or you're resistant to a toxicological outcome? That's one side of things. So we're going now from genomics, functional genomics associations to now genetics, and then finally going down into using models like Drosophila melanogaster and literally having the hypothesis that this is the pathway of harm, this is genetic variation that's tied to your probability of being harmed. What if we manipulate those genes? And that will be our smoking gun. We can't do that for 250 compounds, of course, but at least it will demonstrate the fact that that whole process, that whole discovery process does lead to a variety of different biomarkers that have different levels of certainty. But when you get down to that level, you are very, very certain that when you see something genetic, you will be able to predict something phenotypic. And it requires these genetic modifications to do that.
Yeah, no, it's great indeed. You tackle a good point indeed, that these sort of models, they don't give you just the power to do something, but also kind of the finesse of going where you want to go and not, mess up everything around, because these things are extremely complicated and I cannot stress it enough. And I want to encourage everybody from home, if you have any questions or comments, you see our guests are very eager to reply to all your questions. Now we talk a little bit about PrecisionTox. I hope people had a better idea of what they are trying to achieve, and they are impressed as I am right now. But now I'd like to give to everybody a glimpse of what actually working in PrecisionTox is and all the different people that are around, because I was lucky enough earlier this year to go to the General Assembly meeting of PrecisionTox. So everybody came together to discuss for a couple of days in Barcelona, and we shot a short video, and now I'm going to show it to you. There are also a few of our guests in the video, of course, and then we come back and we can discuss again a little bit on that. So we see each other in a couple of minutes. This time we are flying to Barcelona for the General Assembly of PrecisionTox, where all the members of the project meet to see what they did in the past year, eat way too much paella, and play some volleyball on the beach.
I'll be reasonable today.
5 minutes later.
introduce you to a hero.
How many genes does Daphnia magna contain?
We are trying to proposing a framework surrounding Daphnia as a sentinel species. We're going to unlock different possibilities along the regulatory path.
Basically, what we're doing is trying to create custom systems to analyze fruit fly behaviors.
Yeah, I think one of the interesting things was actually to understand the comparative toxicology part of it. Yeah. So because we are usually focused on one model species or maybe two, and then to understand how the other ones really behave. For example, I didn't know that Daphnia had a locomotory imbalance very early on with casein. Yeah, and so these were some lessons learned.
I really felt that we were belonging to something that was really bigger than just our lab and the work package, to have everything and we see how we fit into that big project.
You need to have, in these big consortium, you need to get the people physically together.
It was really cool to meet just the people who do the same work as you and have the same problems as you, and we're also getting frustrated. Yes.
Yeah, I think it's really helpful to see how we can use other people's knowledge and resources in our own work and keep collaborating and keep building on things together.
I'm looking really forward for this project. It's a real privilege to be part of it.
Okay, I hope you liked it.
I loved it. Well done.
Well, it was thanks to the subjects rather than the shooting. But I hope this also gave everybody a glimpse of how many different things are part of this project, because here we also have a limited amount of time to speak about the project, but there are so many moving parts together. And I'm glad that I had the time to remove my sweater. It was so hot in here at 22 degrees in my studio. But anyway, the question that I have after this video, because here we see that there are so many different parts coming together, it's kind of a puzzle to put together. How easy it is, in your experience, working in this project with so many different expertise? What are the advantages and maybe the disadvantages of doing this? And this time I'm going to start with Gaelle, because otherwise you're always the middle one. So I'm going to start with you, then we go with Christina, and then with John.
Okay, so I would say that for me, it's really a big discovery, because I had never worked on such huge project before. And I would say that what I feel the most about PrecisionTox is that it's so cool that we are able to interact with so many different people that works on so many different that have so many different expertise, and that everything can just come together and we do this one very cool thing all together. So yeah, I think that's really how I feel about that project.
Yeah, no, it was also the impression that I had when I was speaking with people there. They really, almost everybody I would say, is very invested in what you're trying to do. And for you, Christina?
Yeah, along the same lines. I mean, I also never worked in any of the big projects, or an EU project, which is a very different scale even to other partnered projects. And you have a lot of ideas and a lot of experts and a lot of really amazing discussions, and so many possibilities for cross-training, or just getting to know a different model or a different system, or how does that work for you. And you can just jump on a Zoom call, have a 15-minute chat, and you suddenly know 5 new things and can adapt something, or something else moves along. And the possible only downside to this is that you have very long meetings, because everyone has lots of ideas and lots of opinions, which not a disadvantage per se, but it does take a lot of time. So
I can imagine.
It's amazing, but it takes a lot to coordinate. But we have fantastic project managers, so organization is a lot to do, and I'm very glad it's not my job.
And for you, John?
I agree. We do have a superb team. The project management team is amazing. I think that the greatest thing about PrecisionTox is really, while we're looking at it, it's the young investigators within the consortium. They're the ones who basically are bringing life to the whole project. Project conceptually sounds great, but it means nothing unless we have that kind of devotion to seeing it through and the talent to make it happen. You know, one focus that we have in PrecisionTox College is very much on promoting people's careers and making them grow scientifically and personally through this experience. And it fits with our general philosophy. The whole project is really meant to not necessarily just capture lots of data, but to change our minds and people's minds around what's important and what to look for. Kind of like to break down barriers, whether it's breaking down traditional barriers on the path to a person's career, to breaking down traditional barriers and separating ecotoxicology from human toxicology. The whole idea is to try to reinvent the way that we approach solving problems. And it's not just a scientific reinvention, it's a sociological reinvention. You know, how the consortium grows as a consortium feeds into the scientific process and vice versa. So it is very much of a holistic project. It requires a lot of talented people to see it that way and to appreciate it for what it is. And at the end of the day, what success looks like, there's going to be a lot of people who would have learned a lot from doing this project, a lot of people who would have learned a lot from seeing the outcomes of this project, and possibly even some changes in the way we traditionally do things as a result of training people, of demonstrating different data sets or different ways of capturing toxicity and regulating around it.
Yeah, no, I understand. It's also the idea of this such an ambitious project. Indeed, he wants to also leave something after. And the legacy, one of the legacy would definitely be all the different scientists like Christina and Gaelle that will come after this. Okay, now I think I took enough of your time for now, and there is always a moment in our show in which we try to step a little back from all this very serious discussion that we always have and try to have a little fun. And that means that I'm going to step out from the studio for a few moments, and I'm going to leave the stage to Leonie, which is my colleague, that will bring a little fun, or she's going to try to bring a little fun into discussion. So I'm going to leave the stage to Leonie after just a short commercial break of AlterTox.
AlterTox presents DataBox, the first game about new approach methodologies. You want to be part of the journey towards alternatives to animal research and tests? Change is one game away. Come and embody a scientist on their path to develop a new NAM. Tile by tile, step by step, play your way towards validating your method. Save animals while creating your path. And understand the needed steps for validation and dissemination. Play, learn, raise awareness, discuss, teach, support. May the fun be with NAMs.
Hi everyone.
Hi Leonie.
Hi, Leonie.
I love that Matteo always introduces me as the person who brings the fun. I'm the only German at AlterTox, and I don't know why this role of bringing the fun came to me, but I try my best. I like a challenge. So I'm very, very happy to have this little time with you tonight. And I want to bring a little bit of fun. I want to talk about, John, your favorite model animal. I want to talk about Daphnia for a little bit. And I created for the people who are watching us at home. Oh, there's a little critter. I prepared a little quiz for us all here together in the studio, but also for the people at home to join us. So if you want to get your devices ready, you can log into this Slido via the QR code, or just go to slido.com and put in this little code that we have. And we can talk a little bit about Daphnia and get to know them a little bit better. So I want to jump right in, come to the first question. And John, you actually mentioned this at the beginning of your talk with Matteo. So we can also check if people actually listened and can answer the first question. What are daphnids? Are they crustaceans? Are they insects? Or are they their own taxon and not closely related to anyone else? So basically aliens to our planet. So we can see if people at home join and give us the right answer for this question. But we can also see Gaelle and Christina, I assume, even though you come more from the in vitro side, that you know which taxon they belong to. Okay, that is great. Okay, so maybe we can check if there were some answers to this question, Matteo. People listened, John. It's amazing.
Wonderful, Leonie.
Yeah.
Oftentimes, being called a water flea, people would imagine that it's an insect, but it was called a water flea a long time ago because of its pointed nose that you see there, that rostrum. And when it's stressed in the environment under low oxygen, it produces hemoglobin. So it's already an indicator species for low oxygen because it would be shining up bright red because of that state. And people thought, oh, it's a bloodsucker. So therefore it's a water flea, but actually not. It's a blood maker.
Yes, and it holds a very important spot in ecology. So it has been a model not only to toxicology, but also for ecology, because they belong to the zooplanktons, so they play a very crucial point in the food web of lakes. And from their presence, their abundance, and also their appearance, you can already determine a lot about the status, the presence of predators to daphnids and so on. Because some of them, I don't know which species exactly, they form this little spiked cap as a defense when there are certain predators present. So they are all around just a very, very interesting model organism. Okay, so then let's move to the next question. We'll get a little bit more challenging, probably not for you, John, but maybe to some people who come more from the toxicology side. So what is so special about the reproduction of daphnids? Is it that they only reproduce after a specific audio stimulus, and no, not very wide? Is it that they reproduce asexually under normal conditions and sexually when stressed? Or is it just a normal kind and stop being weird? So maybe we can jump right ahead to see if we have some answers coming into this question. And yes, people apparently know their daphnids pretty well out there. So maybe, John, I don't know if you want to describe a little bit what you can do also with this type of phenomenon with daphnids.
I'm impressed that there's 100%. We have to have a very educated crowd here about Daphnia. The cyclical parthenogenesis is really important for our work in PrecisionTox. Cyclical parthenogenesis, as you said, Leonie, means that they reproduce both sexually and asexually, and they cycle around those two modes of reproduction. So when stressed, when not stressed, female Daphnia will produce genetically identical daughters, and lots of them. So we can have whole populations of individuals that all have the same genome. So they're genetically all identical. But when they do become stressed, then they'll switch from producing genetically identical daughters to genetically identical sons. So no sex chromosomes here. This is all environmentally induced sex determination. And then they would require then mating between the males and females to produce now genetically distinct eggs because of meiosis. And those eggs would then go into some state of dormancy. And when they break dormancy, when they return to being moving organisms, they are all genetically identical, and then all genetically unique, and then they will start reproducing asexually in a population, growing the population very rapidly.
Yes, also a very interesting trait that they have and that they use to adapt to stress in their environment. If conditions are not favorably, if they're stressed by just their environment in general, temperature or availability of food or water, they can go and change their complete reproductive cycle to adapt to that. And that's also the basis of OECD validated tests, the 21-day reproduction test with Daphnia, where you go ahead and check, do they reproduce male offspring as an indicator for stress in their environment, and you can use that to do ecotox testing. And then
Leonie,
I think you already,
Leonie,
yeah.
As good scientists, I have not yet tested the effects of berry white on Daphnia reproduction. I think we should try that in the laboratory.
Yeah. And I think you already jumped ahead into our next question, but we can still see if people are listening. So what is the special characteristics of Daphnia eggs? They have a disposition for sibling egg cannibalism. Their coloring differs according to surface they are laid on, or they are dormant and can be resurrected from this dormancy even after hundreds of years. And I assume people listened when you talked, John. So I also assume that we will have, again, 100% correct. This is a very nice test to take. So maybe we can also talk a little bit about the possibilities of that, because when I heard about that, it completely blew my mind, because people also use that to do environmental testing, that they use dormant eggs from sediment core samples of lakes, where you can, I don't know if people have seen this before, you just take a core sample, so you go back in time through the sediment layers, and if you find these dormant eggs in very old sediment layers, you can then resurrect them. If you bring them to favorable conditions, you can bring them back to life and run tests with them and compare if these very old populations react different to modern populations from the same area, for example, to environmental stressors such as chemicals. And I love that this field is called resurrection ecology, which is also just sounds like super much fun. We have some rebels who said that the coloring differs. So not 100% on this, but most people agreed that, yeah, the eggs go dormant. So John, I don't know, did you, or also Gaelle, Christina, did you hear about, of these experience before, that Daphnia eggs can be resurrected?
Yeah, so actually I heard about it during one of the PrecisionTox meetings.
Same for me.
Oh,
I think it was Louisa Orsini who was talking about it. So we learned by being in PrecisionTox.
Perfect. It's, yeah, just such a great source for knowledge around these model organisms. And I'm just, so the study with the oldest resurrected eggs that I know of, the eggs were 700 years old. So that's really predates even the glimpse of the beginning of the industrial age. So we can really use the specific traits of these animals, our understanding of them, our knowledge of ecotoxicology and evolution to get a better understanding on how environmental pollution formed also the evolution of organisms. And this, for me, so John, same, I'm very fond of Daphnia as a model organism. We have them in the labs at the university that I work in Aachen as well. I've worked with them a bit, teaching to young students, and it's, yeah, all over. When people work with Daphnia, they fall in love with them. And for everyone, just like John, like next time you're on a lake, or even if you just sit by a pond, they are everywhere. They're very resilient animals. They're a great model. And I feel just like Drosophila and C. elegans, they're just one of the very big celebrities of model organisms. So very cool animals overall. It's very good.
Agree.
Yeah.
And that 700-year-old clone is very well and alive in the basement of some building on University of Birmingham.
Are they?
Mm-hmm. So the yeah, once you have them, you can keep them alive indefinitely. So you have a whole population of Daphnia going back through time. So we don't have to infer evolution. We can do experiments with evolution. And yes, that is indeed some work of some very cool people at the university. It's
amazing. It's really cool. So also, I want to just say thank you to all of you for also bringing some new information for me about PrecisionTox. I know the project for some parts, but for example, I did not know that there's this long-standing history that predecesses the beginning of this project, and I'm very interested to see where it goes, what we find out, and what we can learn more about Daphnia in the future. So thanks a ton for doing this little game with me, and I will give back to Matteo now.
Bye, Leonie.
Bye.
Bye, Leonie.
Okay, thank you, Leonie, and thank you, John. Now I'm gonna definitely have nightmares about Daphnia, thinking that they are in the basement of my building or something like that.
Matteo, people are going to think that this project is all about Daphnia. The key thing about the project is that it includes Daphnia, but it does also have, the embryos of Xenopus, the clog frog, the embryos of Danio, the zebrafish. And the beauty about this project is that, every one of those species are really unique in terms of their being models.
Mm-hmm.
But when you bring them all together, and what we do is that we stress the idea that we're not extrapolating from a species to humans, let's say. What we're extrapolating from animal biology to humans, because humans are part of that animal biology. So it does require some experiments and data that has all 5 of those model species plus human cell lines to basically complement the whole picture. So it's really the complementarity of that data. As somebody said during your video, it's the comparative aspect that is driving the motivation for this project.
No, yeah, it's a great point to make, and they all look very bizarre to me anyway. What I have in mind every time I see the poster of PrecisionTox, but I understand the scientific rationale behind it. Okay, I took pretty much all the time that I had for you. I have just one question, which is maybe the most difficult one I might have for you all, which is what is next after PrecisionTox? I know that maybe I'm jumping ahead a lot of years, but what is going to happen after? What do you hope is going to happen? What do you hope PrecisionTox is going to bring? Both from the toxicology point of view, but also from your personal point of view. You are free to apply whatever you prefer. So maybe we start with Christina this time, then Gaelle, and then we close with John.
Okay. So, well, the biggest thing basically for me when it comes to what would be great if it came out of PrecisionTox is just for it to have actual staying power, to actually create something, have a toolbox, have something at the end of it that is doing good, that is actually doing something that is usable and is used and helps to actually go into legislation. So that would be my biggest hope, what comes after PrecisionTox or comes with PrecisionTox, that there's a change that comes with it. And for me personally, I will not even be still in my PhD before the project ends. So
I never say never.
Concrete plans yet, but we'll see. There's a lot of things I can still explore and want to explore, where PrecisionTox is definitely going to be a help and just facilitate opportunities for that, to get some cross training in, to visit some other amazing people in the project and learn from them and see where that takes me from there.
That's a wonderful answer. Gaelle?
Yeah, so on the first part, I completely agree with Christina. What I hope for PrecisionTox is that it will have a real impact for society, that we will be able to create that toolbox that will kind of change the regulation for the better, and also maybe help the EU to reach their project of no toxic chemicals at some point. So yeah, that's really my hope for PrecisionTox. And then for me, I think that this project will also help me to go on with my career after. So again, I hope at least. We will see.
I'm sure it will. John?
And Matteo, thanks to your show, it's already proving to be quite giving in terms of outputs. There's a nice message from Chiara saying that today we have a new person intrigued about Daphnia. So already we're succeeding.
Sorry about that, Chiara. I take responsibility.
But again, 2 things. One is definitely to ensure that there's a successful career for those who are who are participating in it. And through that career development, we have more and more of PrecisionTox gaining traction in all kinds of places, industry, government, academia. But also importantly, it is a very unique concept. And if we can definitely show that the concept is true, that we can understand the human condition better around toxicology because of knowledge that's derived from this type of investigation, I think that the outcome will naturally progress into changes in the way we do things like regulation. But it's going to take time, and PrecisionTox doesn't rely on science alone to make that change. There is significant work within PrecisionTox that involves not scientists, but legal experts, lawyers, looking for those early wins, the socio-technological barriers that are holding back NAMs. As we said before, science is only part of the solution, and PrecisionTox hopes to capture as much of the complexity of the problem as possible, so to address it holistically and not just focused on how Daphnia responds to chemicals, for instance.
Well, I think you made all a great job to painting how this project is working, and I thank you very much for your time. I have used a little bit more than the time that we agreed upon at the beginning, but it was very interesting for me, at least. I think a lot of people indeed thought the same. But again, the project is so big. I cannot stress enough how many things are happening in PrecisionTox. So if you are interested, please, you can go and have a look at their website. There are so many more information you can find there. You can also find the contacts of other people that you can contact if you want to know more. As I said, PrecisionTox is not alone in this huge endeavor. There is also other projects that are working together. One is OnTox, we spoke about a few weeks ago. Riskanter is the other one, and they are all merged together in this huge cluster called ASPIS. And again, you can have a look of what they are doing there. It is truly amazing, the science and the what all these people are trying to achieve all together. It's impressive, and everybody should have a look at it. And with this, I thank you very much for your participation, John, Christina, and Gaelle. I wish you all the luck possible for the project, and I hope to speak with you soon.
Thank you, Matteo.
Yeah, thank you for this evening.
See you soon.
Yeah.
So that's the end of the last episode for this year, at least, of TOXstreams. We are going to take a break for Christmas, and we're going to be back at some point in February next year. So stay tuned on our social media to know exactly when we're going to be back and what we are going to discuss about. For the time being, I thank you very, very much for your participation. I hope you had fun. I hope you learned something during this episode, and I can't wait to see you back next year. So see you soon.